Well, good morning. I'm Laurie Glimcher. I'm the Dean at Weill Cornell Medical College and am the moderator for this panel this morning. And so thank you, everybody, for joining us for a panel on cancer immunotherapeutics. Our four panelists today are going to focus on the role that new biologic drugs that target immune receptors, so-called checkpoint blockers, as well as vaccines against cancer and cell-based therapies, so-called CAR T cells, what these new approaches, how they work in mobilizing the immune system to become activated and kill tumor cells, are doing. And you're going to hear a lot more detail from them, so I'm going to make this introduction pretty brief. I don't need to tell any of you how devastating cancer can be, and probably most of us here have experienced that at a personal level or in families or friends. Currently, 4% of the United States population, that's nearly 14.5 million people, are diagnosed with cancer in their lifetime. That's triple the percentage we saw 40 years ago, and the most recent estimate from the National Institutes of Health indicates that the overall economic cost of cancer in the US is about $216 billion. That figure is only going to grow as our population ages, which it is doing rapidly, and the number of people dying from cancer is going to increase from 8.2 million in 2012 to an estimated 14.6 million in 2035. The vast majority of these deaths, of course, are not due to the primary tumor, they're due to metastatic disease. Immunotherapy, which is what we're going to be talking about today, has recently exploded on the scene, as all of you know, and I think will prove to be one of the most promising and exciting areas of investigation into cancer. It may also be useful for other diseases where the immune system is suppressed, such as HIV/AIDS and other viral diseases. Now, we've seen over the last 5 years a series of major successes. In 2011, we introduced CTLA-4 specific monoclonal antibodies. CTLA-4, as you'll hear more from Dr. Bluestone, is a negative regulator of T-cell immunity. And this first agent, which was called ipilimumab or Yervoy, was produced by Bristol-Myers Squibb. It was the first agent to show a survival advantage in patients with advanced melanoma. A very profound effect on metastatic melanoma. More recently, antibodies to another immune receptor called PD-1 receptor or its ligand, suppress, which is also an inhibitory receptor, have been developed, and that includes an FDA-approved drug from Merck called pembrolizumab or Keytruda for melanoma, and experimental agent nivolumab, also called Opdivo, from Bristol-Myers Squibb for both melanoma and lung cancer, and you're going to hear more about that from Dr. Giordano. Recently, there have been 2 studies that have showed for the first time that adopted T-cell therapy for patients with advanced melanoma can cause tumor regression. Steve Rosenberg at the NCI had similar results in patients with melanoma and patients with metastatic synovial sarcoma. Most recently, there's been groundbreaking work on what is called chimeric antigen receptor therapy, or CAR, and that has occurred in 2 laboratories primarily. The first is Carl June at the University of Pennsylvania, and the second, Dr. Michelle Sadelain at MSKCC, and you're going to hear more about that during the panel discussion. And these have been clinical studies performed in patients who have chronic lymphocytic leukemia or acute lymphocytic leukemia and have targeted a B-cell antigen on the surface. You'll hear more about that, and we'll talk about the promise and the challenges of that therapy for our patients. to recall that although we've made good progress in treating some cancers, we still cannot treat many of the solid tumors that we see in the clinic. So we still do not have adequate therapeutics for tumors like pancreatic cancer, ovarian cancer, lung cancer, and so forth. And as an immunologist, I have witnessed these enormous breakthroughs with much delight, because this is a field that started a very long time ago and really represents a triumph on the part of basic immunology, because they all stem from our ability to elucidate these key immune system molecules and their function. So I think it's very important to remember that these advances stem from over 100 years of research, and that the field of cancer therapy has now benefited from the work of dozens of investigators across a spectrum of disciplines. This really began in 1893 with William Coley, who was a New York surgeon, and he first noticed that some cancer patients seemed to be helped if they got an infection following surgery. So he began treating them with a mixture of live bacteria and bacterial lysates that were called Coley's toxins. And this eventually resulted in the treatment of bladder cancer with BCG, which is an adjuvant made from M. tuberculosis. And that met with some success, was followed with the advent of other forms of treatment like radiation therapy. So here today, we have 4 expert panelists who are going to discuss these recent breakthroughs and future directions in the field of cancer immunotherapy. And I want to introduce them to you all at once so that I don't have to keep on coming up and introducing them. So let me do that. Dr. Jeffrey Bluestone is the A.W. and Mary Margaret Clausen Distinguished Professor of Metabolism and Endocrinology. He's director of the Hormone Research Institute at the University of California, San Francisco. Jeff Bluestone is one of the world's leading immunologists. He's been a pioneer researcher in the fields of T-cell activation and tolerance, and helped develop the first FDA-approved drug targeting T-cell co-stimulation, about which you'll hear more in a minute, to treat autoimmune disease. He was also the first to demonstrate the checkpoint inhibitory function of the T-cell activation molecule CTLA-4. And that really helped to usher in a new era of cancer immunotherapy, leading to the first FDA-approved CTLA-4 monoclonal antibody, Yervoy, that was used in the treatment of metastatic melanoma. Jeff's recent research has focused on the critical role of another kind of T-cell called a T-regulatory cell in immune tolerance, and he has spearheaded and is in the midst of several clinical trials testing the role and ability of T-regulatory cells to treat autoimmunity and organ transplant rejection. So he's opened up a new field of cell-based immunotherapy there. He's published over 400 papers, has received numerous awards for his work, including election to the American Academy of Arts and Sciences and the Institute of Medicine. He's received a Guggenheim Fellowship and a Merit Award from the NIH and a Distinguished Alumni Award from Weill Cornell Graduate School of Medical Sciences. So that's Dr. Bluestone. Dr. Robert Vanderheid is the Hanna Wise Professor in Cancer Research and Vice Chief of Hematology and Oncology at the Abramson Cancer Center and Perelman School of Medicine at Penn. Mike, you mispronounced that. It's Perelman School of Medicine at Penn. And Bob combines vigorous efforts in both laboratory-based and clinical research. He led the development of telomerase as a universal vaccine target in cancer, and he'll speak more about that, as well as antibodies against another co-stimulatory molecule called CD40 that activate them. He has studied a number of cancers, including melanoma, breast, and pancreas, and most recently has co-led a Stand Up to Cancer Dream Team initiative that looks at the combination of several of these therapeutics for pancreatic cancer, one of the most difficult cancers that we have and still cannot treat. Michael Giordano, raise your hand, thank you, is a physician who is Senior Vice President and Head of Oncology Development for Bristol-Myers Squibb. His leadership in drug development over the last 15 years has brought several innovative medicines to patients in areas such as HIV, hepatitis B, and most recently, oncology. He's been the leading contributor to the introduction of immunotherapy into the oncology treatment landscape. Michael led the development of the first checkpoint inhibitor, we've already mentioned, ipilimumab, or Yervoy, and he currently leads a very broad portfolio at Bristol-Myers of immuno-oncology and oncology agents that offer the potential to redefine our cancer treatment. And finally, Stephen Stein, who's a physician, is Senior Vice President and President of US Clinical Development and Medical Affairs at Novartis Oncology. Dr. Stein's primary responsibility is to lead oncology medical and scientific activities, including the planning, the resource allocation, and execution of registration and non-registration studies in medical affairs, clinical programs. He's also responsible for the medical science and health outcomes function, which includes both scientific operations, scientific communications, medical information education, and health economics and outcomes research. So we have an extraordinarily talented panel of experts here to discuss this important area. And I've asked each of our panelists to provide you with a very brief introduction. And I've also asked Dr. Bluestone to show you just a couple of slides on the mechanism of actions and the molecular mechanisms that underlie these extraordinary advances that have been made. Okay, thank you.
Thanks, Laurie. It's a pleasure to be here and a pleasure to talk to everybody today. I guess people can hear me now. And I don't want to take too much time because I suspect a lot of people in this audience have read a lot about the revolution in cancer immunology. But I think it's important to take us back a little bit in time to think about what the field looked like 25 years ago, when after decades, and as Laurie mentioned, many decades of attempts to try to use the immune system or even think whether the immune system could be used to treat cancer, there was a great deal of skepticism, frankly, about the opportunities that were present. And I think the skepticism turned out to be a consequence of the biology. The biology of the immune system is really focused on preventing the body from being able to eliminate cancers. And on my first slide, the person, is it up? Oh, no, that's my second slide. Okay, so the way I look at it is, you know, the immune system is a body at war, right? At the same time, we're trying to mediate an immune response against the things we want, virus, bacteria, and the like. We are desperately trying to control inappropriate responses against our self-tissues, whether it be our pancreas and type 1 diabetes or our joints and rheumatoid arthritis. The ability to be able to control that in most of us in this room is critical to the viability of humans. And so systems have been put in place, systems, backup systems, and backup to the backup systems, all designed to shut down inappropriate responses against self-tissue. And so what happened was, starting in about the mid-'80s, we started to understand all of those mechanisms that were put in place. Some of them were cell intrinsic. We would have molecules on the surface of cells that needed to be activated, in T cells in particular, molecules like CD28, the co-stimulatory molecules, that unless they were triggered at the same time that an antigen was seen, the immune system didn't respond. There were cytokines that had to be produced that were necessary to boost an immune response, but equally cytokines like IL-10 and TGF-beta, which shut that immune response back down. And I think most dramatically was an understanding in the mid-'90s that there was a whole series of molecules that were going to be placed on the cell surface that had the precise role to shut down immune response. And now what we call checkpoint blockade, where in fact this series, as turns out, of negative regulators that are all designed to turn things off. And so the final key was the fact that the immune system not only employed intrinsic regulatory cytokines and molecules on the cell surface to control, but built whole sets of cells. Now we then called suppressor cells, we now call them regulatory cells, that are all designed to be the final backup and keep things in check. And what you'll hear in the next hour or so are how all of these play in to now the new series of drugs that we have been developing to combat cancer. Developing drugs that will boost the immune system by either cytokine or activation of co-stimulation, will reverse the blockade caused by these regulatory molecules like CTLA-4 and PD-1, inactivate the regulatory cells so that they don't sit in the tumor site shutting down any immune activity, and to release what turns out to be a rather potent T-cell response against tumors that, if in fact allowed to proceed, will recognize either common proteins expressed on those tumors, somatic mutations that come up in those tumors, or the like, modifications of those proteins so that an immune response can be generated to eliminate it. So that's the background, that's the context of this. The last point I want to make, and then we'll move on, is that the risk that comes into mind. Remember, all of these methods of control have been designed to avoid autoimmunity. So the other thing that we'll talk about today is the consequence of unleashing this immune response that would otherwise be well controlled, and how we're going to deal with the off-target, on-target for the drug, off-target for the immune system, consequence of unleashing this immune response in terms of autoreactivity, in terms of destruction non-specifically due to cytokine storms that exist from activation of cells and the like. And this is where this whole conundrum will lead to this last topic, which is, can we engineer our immune system? Can we engineer the T cells so that we can maximize their ability to recognize something on the tumor while minimizing the activity that might cause an uncontrolled autoimmunity? And that's where CAR T cells and other modifications of cells come into play. So with that introduction, we can turn it over to any of my colleagues.
Second slide.
I think along the way I might.
Okay.
Okay.
Good.
I'm going to throw up a slide here and there as my colleague starts talking about some of the specific things so people can see a pictorial of them, because immunology is a field full of jargon, if nothing else, and it's hard. Heart disease seems so much easier to understand.
It is. It's still the NCP guideline.
You know, you lower, you raise, it's easy.
I might say a few words, really to tell you how I arrived at where I am today with regard to working in this incredibly exciting field of immunotherapy for cancer, and to build upon what Laurie said about really cracking the code on better immunotherapies for cancer. Really will take a multidisciplinary approach from multiple areas of expertise. I'm actually trained in infectious diseases and spent a great deal of my early career on infections of the immunocompromised host and the use of immunotherapy to treat viral diseases. We didn't get very far, and however, was delighted to see that the HIV therapeutics evolved from a uniformly fatal disease to one which in most instances in developing countries is controlled. That's something that we have all, as Dr. Vangelos mentioned last night, have seen over our lifetime. To put the context of the excitement that I have about immunotherapy for cancer into perspective, I think that this is the first chapter in a very similar story that's likely to be told for cancer on the introduction of this modality, a new modality, to treat cancer. And there have been some very significant points of evidence that some of the modalities to enhance the immune response or to attenuate the negative immune signals are successful. Now, there are many modalities, from vaccines through checkpoint blockade to bispecifics to CAR T cells. And what I'm going to be speaking about mostly today is some of the experiences that we have that are really what I would say not only proof of concept and principle, but proof of survival. So in my view, there are several of the modalities that have had clear demonstrations of survival advantage, and Laurie mentioned checkpoint blockade with CTLA-4 inhibitors, and also now PD-L1, PD-L pathway blockade has been associated with a survival advantage in melanoma. Those successes must get us all excited about driving a deeper understanding of why the therapies work in some, why the therapies don't work in some, and why they work incompletely, and how we can move this to the next level of, maybe you'll show the Walter Erber, what I call the Walter Erber slide, which is this one, which is the one we show almost every day in our research organization, which is how do we move the survival curve, which we've now achieved for melanoma, of 20% of patients achieving long-term survival with CTLA-4 checkpoint blockade, to 40 or 50% with PD-1, PD-L1 blockade, to 70 or 80% with combination therapies. That's the paradigm that I think we need to put on the table for all of us to try and discuss and achieve how we can get that across tumor types and that in the vast majority of our cancer patients. So that's the context with which I wanted to have today's panel discussion with really esteemed colleagues.
Keep marching down the panel. So by way of disclosure as well, I'm not an immunologist either. I don't even share your background in terms of infection. I'm a hematologist oncologist.
That's an okay thing.
That's an we'll see. Trained at Penn and work as a drug developer in a large pharmaceutical company which has a passion for developing drugs in both the liquid tumor, solid tumor, and rare disease space, including endocrine disorders. So just by way of additive comments, what we think we can bring in terms of large pharma, in addition to what Michael said, if you look at our own history, we announced the collaboration with Carl June and Penn in August of 2012, moved very quickly for what we saw was an early and extremely promising and exciting signal. Where we stand today, and we'll talk more about it later in terms of CAR T cells, is have open phase 2s in collaboration with the University of Pennsylvania in acute lymphocytic leukemia in adults and in the pediatric population. That is underway under the Penn IND. We hope to have the Novartis IND open by the end of this year, and have told the world publicly that we hope to file for that indication in 2016. And we have additionally acquired cell processing and manufacturing facility in Morris Plains that has the scalability necessary to manufacture for both the clinical trial program as well as the initial indication. Additionally, in February of this year, the company acquired a company called Costim from Cambridge, Massachusetts, which has numerous molecules in the space, including PD-1, and very recently announced a collaboration with BMS with your PD-1 drug in 3 different spaces, in collaboration with our ALK inhibitor, ceritinib, in ALK-positive patients after progressing on crizotinib, in addition with a MET inhibitor that we develop in, an Incyte compound INC280, as well as an EGFR compound that we have, EGFR 816, that actually hits the T790M mutation. All 3 areas in non-small cell lung cancer that we think hold tremendous promise. So I think just to end on a comment from me, what we can bring is the ability to combine in this area, and as your slide points out, combinations will hopefully get us up to the next point on the curve, which is where we all want to be. Thanks.
My name is Bob Vanderheid. Laurie, thank you for including me in this panel. Real privilege to be here. A few introductory comments, and then we'll get into the discussion. So my perspective is one of a medical oncologist. I take care of patients. I also run a discovery laboratory at the Abramson Cancer Center at the University of Pennsylvania. So I take care of humans, and I take care of mice as well. It's a
Which
nice,
do you prefer?
It's
No answer to that?
of course taking care of patients is a privilege. There's no higher privilege. The points I wanted to make are, to reiterate, this is an amazing breakthrough. We saw that on the cover of Science, declared so in December of last year. It's a 20-year overnight sensation. It's based on science. It's not based on alchemy, but it is based on these amazing clinical results. Our patients, a 7-year-old girl who is now 10 years old, free of leukemia, when she was hours from death based on CAR therapy. A woman I take care of who received an antibody against CD40 for her metastatic and refractory melanoma, who was alive, free of disease 9 years later. I treated her in 2007, last time she received anything. And we're at the, it's not just these past experiences where we're going. We're on the brink of looking at metastatic lung cancer. The trials are underway to displace chemotherapy as frontline therapy. We can imagine the day where we use immune therapy as our first choice, not our backup choice. And that's because of the promise of immune therapy. This slide, I'm glad it's up. The difference between the purple and the blue is very typical in oncology. That's the banana trial. You know, push that banana to the right. And as I've heard my colleague Carl June say recently, you know, we're after tails, not bananas. We're after the green tail. We're after increasing, and that is the promise, and now being delivered by the immune system, which is to cure cancer and watch people walk away from their disease. So what is the basis of this? And I'll just make a couple of points and then we'll get on. The mechanism of action that we're pressing upon is, in large part, a T lymphocyte, a T cell, not antibodies. And this is surprising because think of the last great immune revolution. These were childhood vaccines. These were vaccines for childhood infectious diseases. None of them, now you can correct me if I'm wrong, Michael, but I'll go ahead and put it out there. Not a single one of them was based on the T cell hypothesis. It's based on antibodies. That didn't work in cancer. It might yet work, but it hasn't worked yet as a vaccine strategy. T cell therapies are working all of a sudden for cancer. Remarkable. This is a whole new type of modality that we've never tried before. What are the promise of T cells? That means they recognize a target. So the target had to exist. It took 20 years to understand that. T cells are regulated. They're so powerful. We had to understand the regulatory mechanism. That's happened. We know their names now, some of them. And T cells remember. So you can get a T cell therapy and it lasts. That's why some patients are free of disease. Now, what that puts us in a position is that as we celebrate all these remarkable achievements is that we can't rest on our laurels. In fact, we have an opportunity not to blow it, and that's everybody. That's academics, that's our medical institutions, that's the National Cancer Institute. We're on, and pharmaceutical companies. We're on the brink of something remarkable. Let's not become shy. Let's stay bold. We also have to recognize that not everybody is helped. If you're a patient with metastatic pancreatic cancer, and these are patients I see, there is no promise from anti-PD-1. There is no promise yet reported for anti-CTLA-4. Those therapies do not work in pancreas cancer except for a couple of patients, perhaps. We have to figure out what are the resistance mechanisms at work. This was the fundamental thing. As important as Gleevec was, even more important was understanding why Gleevec didn't work in certain patients. And then we found new drugs, new pathways that actually were even better. So what are the, sorry about that. So what are the pathways? And we're turning to, we know that there'll be more checkpoint blockade that are related to T cells. But we're now beginning to confront ourselves with the rest of the tumor microenvironment and the tumor cell itself. So suddenly, although immunotherapy and cancer immunotherapy was operating in this current and celebrating itself, we've now run totally intersected with cancer biology, because immune surveillance is part of cancer biology and always has been. It's not alchemy, it's not out there, some sideshow, it's part of cancer biology. It didn't first make it into the hallmarks of cancer in 2000, but now it's creeping in the latest edition from Hanna and Weinberg. And the tumor cells and the tumor microenvironment have a lot to say in when T cells work and when they don't. This has to do with inflammatory networks in the tumor microenvironment, which we're only beginning to peel away. And I guarantee you there are tons of targets within the non-T cell tumor microenvironment. And also, final point, we're understanding that oncogenes that drive cancer, and I'll put out there the KRAS oncogene, in addition to everything else they do, they also set up this inflammatory and immunosuppressive network that prevents T cells from getting in the first place. So there's a huge amount of learning yet to be done, a huge amount of science yet to be done to uncover new pathways. I think we're at the tip of the iceberg, and we can discuss that today.
Good, thank you,
Fantastic.
everybody. Maybe to step back just for a minute, because the immune system is horrendously complicated. It's one of the reasons I decided to become a skeletal biologist as well as an immunologist. There are only 3 cells in the skeletal system that you have to worry about. In the immune system, there are many, many cells. And maybe we can just outline those briefly. As somebody pointed out, we think of immunosurveillance in the immune system. And according to Robert Schreiber, who's at WashU, who developed the theory of cancer immunosurveillance, all of us every day are knocking off in one form or another these rogue cancer cells that circulate. And most of us are very effective at doing that. And so we prevent the initiation of cancer. And the immune system is very complicated. It consists of a number of different cells and 2 major systems that we call adaptive immunity and innate immunity. And the adaptive immune system are the T lymphocytes and the B lymphocytes. You're going to hear a lot about T lymphocytes today. They are what I call the brains of the immune system, and they are the cells that when they become activated can lead to an effective response against a number of different kinds of pathogens, including tumors. Also can become effective against a number of infectious diseases. T lymphocytes are not a homogeneous population. They come in different flavors. So there are CD4 or helper T lymphocytes and CD8 killer lymphocytes. Both of these are important in the setting of cancer. To make things more complicated, CD4 T lymphocytes are also not a homogeneous population, but can be divided into several subsets of T cells that subsume different functions, primarily because they secrete different cytokines. So we have what we call the T helper 1 cell, which makes a very potent anti-tumor cytokine called interferon gamma. We have the T helper 2 cell that makes cytokines that are important in allergy and asthma. And we have the T helper 17 cell, which is important in mucosal immunity. And then we have a cell that Jeff Bluestone has spent a lot of time studying, which is the T regulatory cell. And that is an immunosuppressive cell that controls, in some ways, the other 3 populations. And so that's your adaptive immune system. The innate immune system is a much more primitive system and consists of cells like dendritic cells and macrophages and natural killer cells, which each also play a role in combating tumors. And that's what we're referring to as the tumor microenvironment, because these cells taken together interact with each other and secrete a number of substances which can be pro-tumorigenic or anti-tumorigenic. So that's the immune system, and I'd like to invite our speakers to perhaps make a few more comments about the science behind this new age of immunotherapy. And Jeff, I believe you have 1 or 2 slides that outline this so you can see it visually.
Yeah, so let me just work our way backwards for a second from this very large system, which you've heard Laurie and Bob talk about, which is complicated, right? Multiple different cell types, multiple different cytokines. And say, if we just think about the one interaction, which is the critical, the brains of the interaction of the T cell with the tumor and with the cells, the macrophages or dendritic cells that are presenting the antigens, what I titled here is the future of immunomodulation is also the challenge of immune modulation. Because if you just look at this slide, you can see how complicated the whole system is. You have the T cell receptor itself that sits on that cell that needs to engage an antigen. And what should that antigen look like? Should that antigen be uniquely expressed by the tumor cell? Well, we'll talk a little bit more. I might show a slide about the number of possible candidates, but in any individual, it's an enormous number of mutations that one might want to target, and are we prepared for that kind of personalized medicine? But the T cell receptor needs to engage, and it needs to engage in a productive way. And so you can see on this slide, every green line is what you need for a productive immune response. And then that's counteracted by every red arrow, which is the attempt, as I said, of the immune system to shut it down. So even at the level of the T cell, when you hear terms like PD-1 or CTLA-4, you're going to hear things like LAG-3, and the nomenclature is enormously complicated. But what you see on this slide is the amazing ability of the T cell to use a systems biology approach to being able to integrate signals that it's getting from the tumor, from the antigen-presenting cells, and from that environment to do it. So for me, when I think about the immune system, I think about it as this integrated signaling complex bag of signaling molecules. And to get it right, we're going to have that right balance of being able to turn on the T cell when we need it turned on, prevent it from being turned off, and turn it off when we want it to turn off. And that leads to the last point I want to make about the immune system, which was touched on by Laurie, and that is, and by Bob, and that is the amount of effort the tumor makes itself to shut down this immune response and escape detection. It does it through modifying and modulating the ability to express these foreign proteins. The major docking protein for tumor peptides is called the MHC molecule, or HLA in human. And many tumors escape immune detection by downregulating or shutting down the precise system that needs to be recognized by the T cell. Other tumors do it by making cytokines like IL-10, which shut down that immune system. And other molecules like KRAS and other tumor suppressor genes will actually regulate the ability of T cells to even enter into the microenvironment. So it emphasizes the complexity of what we're trying to do here. And where does that become tough? Well, we've had a lot of success in melanoma. We've had a lot of success in the liquid tumors, as we call them, the lymphomas and leukemias. But when you start getting to solid tumors, when you start getting to ovarian, some lung cancers and stuff, getting that immune system to get into that tissue, getting it to actually strike down is going to be the real challenge.
Go ahead.
I was going to build in a different direction, which is on the maybe you want to go first on the basic stuff. I was going to comment a little bit on despite the great complexity, which we all appreciate, one of the remarkable things I think that several have pointed to are the clinical successes that we're seeing at the bedside, and how those are going to help us go back to further elucidate the basic biology. So despite the great complexity, this slide could be 10 times more complex, there are 2 or 3 domains in which we are seeing very remarkable clinical effects. And I'm perhaps somewhat biased, but the 2 domains that I think we're going to talk about today are CAR T cells, and others know more than I do about those, and also checkpoint blockade. So within the realm of only focusing on 1 or 2 of the pathways of T cell regulation, either CTLA-4 blockade, ipilimumab, tremelimumab, or the 4 or 5 drugs that work in PD-1, PD-L1 pathway blockade, we are seeing remarkable advances, which is rewriting the book of how you do drug development for a new paradigm, because the way these drugs work is not the way that chemotherapy works or targeted agents work. So there's an entire new drug development paradigm, which as academics, industry, and government, and advocacy groups, we need to really understand that the shape of the curve is not a banana. It's hopefully a tail, and a very wide tail perhaps. So I do want to emphasize a little bit the remarkable observation that PD-L1 blockade, PD-1 or PD-L1 pathway blockade has resulted in clinical activity in more than a dozen tumor types. Now, the range of how good that activity is large, and we're seeing very important results in melanoma. We're seeing very important results in lung cancer. We're seeing underappreciated, and in my view, extraordinary results in renal cell cancer. And we're seeing in the liquid tumor domain, in both Hodgkin's disease and non-Hodgkin's lymphomas, data that I think will surprise all of us by the end of the year. At the same time as we're hearing reports on bladder cancer, ovarian cancer, breast cancer, head and neck cancer. So I do want to, I'm not saying that we have the panacea has arrived, but I am saying that the first chapter is over and the second chapter is here, which is to try and understand how we can leverage the clinical observations that we're seeing in a very large number of tumors and in a substantial number of patients, inadequate for what we want to achieve in the long term, to really drive some of the biologic understanding as well as why we have responders and why we don't. So it's now, I think it's now bedside, back to bench, as we reflect upon what next to do in that slide. That's one perspective.
I wanted to pick up on that point, and also back to Laurie's question about what are the fundamental scientific principles. And Laurie mentioned work by Bob Schreiber at Washington University, St. Louis, who coined this term immunoediting, and starting in 2001, rewired our minds into thinking that, that cancer immunosurveillance wasn't a laboratory artifact. And what he described was basically a Darwinian type of selection process where a malignant tumor or neoplastic cells form. The attention of the immune system, particularly the adaptive immune system, is garnered, and there's a battle royale that happens sometimes subclinically. And if the tumor can escape the immune onslaught, then it emerges. But it emerges as a different type of cancer than it began with. It's edited, immunoediting. It's Darwinian biology in many ways. And what has been discovered is that those model systems that Bob used, elegant work, had underlying those tumor systems were many, many mutations. Those were some of the most mutated tumors, carcinogen-related tumors. And they do look a lot like certain human tumors, sun-related melanoma, smoking-related lung cancer, bladder cancer, which, by the way, are the tumor types leading the way in the highest rates of success with checkpoint blockade. But there is emerging view that it doesn't always work that way. And I think we have to keep in mind that there's another way immune surveillance can become frustrated, and that is rather than the cancer cell drawing the immediate attention of adaptive T-cell responses, that in fact there is immunosuppression from the beginning, and that one doesn't see, there's just an exclusion, almost a privilege, immune privilege, of tumor microenvironments as they are under the influence of, for example, oncogenes, so that there's never an immune response. There's never a T cell there. And when you then look at these patients, you can look early or you can look late. You just don't see T cells in there. Whereas in melanoma, about 30 to 50% of patients with metastatic melanoma have lots and lots of T cells. Triple-negative breast cancer has a pretty sizable amount of T cells, whereas other types of breast cancer do not. And so from an immunologist point of view, we're starting to see there are immune-infiltrated tumors, inflammatory tumors, and then there are the ones that are privileged. And it may be that that we have to treat those types of tumors differently. But I do think there's more biology than the Darwinian biology. There's this sort of privileged biology as well.
So I think that brings up an interesting question, and that's the use of combinatorial therapy. So what really changed the landscape on the treatment of HIV, turning it from a lethal into a chronic disease in most instances, was the realization that you needed to hit the virus with multiple agents at the same time. Now, there are many tumors which you say, as you say, are not particularly, quote, susceptible to immunoactivation therapeutics. And so, and the vast number of therapeutics we have now, of course, hit the tumor directly. So wouldn't the ideal here be to simultaneously hit the tumor directly with whatever agents, whether they're the new targeted therapeutics or whether they're chemotherapy or radiation therapy, and at the same time activate the immune system? And I know that such trials are in their infancy now, where we do combination therapy with agents that directly hit the tumor and at the same time activate the immune system with checkpoint blockade. So maybe any of you could speak to what you think the future of that is, because from what I've heard, there's quite a lot of toxicity when you do that combination therapy. And so how are we going to manage that?
And maybe I'll start, and maybe you, since we have some collaborative work together on TKIs with checkpoint blockade. It's been our thought experiment in our development organization that, in fact, we would like to evolve combinations of immunotherapies as the most likely long-term solution to cancers to replace chemotherapy. However, that will require an increased knowledge of how to make those combinations of either checkpoint blockade, NK cells plus checkpoint blockade, LAG-3 for T-cell exhaustion, and that's in its infancy of understanding. And that is our principal mid to long-term goal at BMS. At the same time, however, we need to do all of the good work to look at whether priming is important with either radiation or anti-neoplastic, cytotoxic or targeted therapies. We've been, to date, disappointed in the combination of immunotherapy, specifically checkpoint blockade with TKIs, because of principally toxicity. Tyrosine kinase inhibitors such as BRAF inhibitors, sorafenib, and a few others, in which we've seen an augmentation of the toxicity, principally hepatotoxicity. But that's something which is in its early days. Nonetheless, there are evolutions in the quality of targeted agents, better ALK inhibitors, better EGFR inhibitors that I think are likely to be part of the armamentarium in some of the very large tumors, which is why we're doing some work with your group, Steve, and maybe you want to build upon that.
So just to go back to the safety issue for a second, in terms of CAR T cells, and not to be the sort of party pooper here, and Bob alluded to it, but there is, with great responsibility, becomes an intense focus on safety, and certainly as we roll it out sort of beyond the centers of expertise, we're going to have to be careful. So for example, with CAR T cells, in this enormously complicated system you explained, cytokine release syndrome has become, a dominant toxicity that is problematic and needs to be very carefully managed, and seems to be, from the literature, related to tumor burden as well. And that seems to be one of the issues that we're going to have to learn how to measure upfront and predict and then manage appropriately without harming the T-cell expansion, which is necessary as well. So, that is something, as we go across the United States and open this up further, and then globally, that is going to be something we're going to have to watch very, very carefully and manage very carefully. And that's, again, talking about monotherapy. In terms of the combinations, I think, you said, to do the iterative experiments preclinically as much as possible, but then not, once we have an idea that it'll work preclinically and that there is enough evidence that it should be safe, then to go out carefully in humans and do the experiments. So the areas, to your point, Bob, that are really mutagenic tumors that we think will work in, and then use an appropriate targeted therapy like an EGFR inhibitor, that third generation now that hits T790M with PD-1, would be the right experiment to do, and then go through those very systematically and iteratively, but very, very safety focused, because again, even with CARs with the enormous promise, there is this safety issue that may become more problematic as we roll it out beyond centers of expertise.
Yeah, I just want to make two comments. One is that we've been talking, and at least the sense I get as we talk here, is we talk about things in one time frame. And I think what we're learning about the immune system in regards to all of these molecules is that at different times during an immune response, there's probably a different time in which these molecules act. For instance, CTLA-4 undoubtedly acts early on when antigen is first recognized, and if the cells don't get a strong enough signal, they shut down. And that means that you're going to have a certain T-cell repertoire. You're going to have a certain type of antigen you can see or not see. PD-1 is likely acts over a longer period of time at the tumor site more, and in fact, you get what's termed exhaustion or anergy, or the cell has too strongly recognized an antigen without the right cofactors and gets shut down. So I think combination therapy, as I see it, is not going to be, let's see how many things we can put into a bottle and inject it all at once. It's going to be being able to understand through biomarkers when is the right time to give the right drug in the right person. And that's going to require animal models, human testing that comes back, and that's where I think we're going to learn a lot more about why people respond or don't. The second point I want to make, and I think it's really important to make it in this room with these people who are sitting here. I've been academic, so I just watch things from the outside. And for the longest time, I ran an organization that tried to do clinical trials in autoimmunity and transplant. I'd go to two companies and I'd say, if I could put your CTLA-4 Ig together with your anti-CD40 ligand, I could induce tolerance, I am sure of it, and never made any progress. And the response was always that the pharmaceutical industry was unwilling to take chances on combination therapies, especially with drugs that were not approved yet. We would destroy their market if we had an adverse event in another. And these two guys sitting here to my right, and what's going on in this field right now is extraordinary, because in this field today, people are combining unapproved drugs, not approved drugs together, and they're combining them in ways that are cross pharmaceutical companies. And I think this is going to speed up the field, and it's going to be an incredible positive for the patients.
And that's true not only for cancer immunotherapies, but also for drugs that target the cancer directly. There's been a lot more collaboration between pharmaceutical companies in combining drugs, and that in one sense balances the risk as well as the benefit.
The catalyst has been a really, I think, in many ways, academic influence, advocacy influence, and so I think that's been all a very good partnership.
So let me ask you, Bob, or you, Steve, to talk a little bit more now about CAR T cells and define them for this audience, and present some of the challenges that we see, because of course they've been tried in very few types of tumors, and initial trials resulted in some real safety issues, which look to be more controlled now, safety issues due to superactivation and the production of what we call a cytokine storm. So why don't you define these for us, and then tell us where you think we're going. There have been some stunning successes in the liquid malignancies.
Okay. I asked Bob if he'd go first and do the whole preclinical explanation, but he refused. So I will go ahead. No, so the prospects are certainly good. The second generation CARs now are with multiple companies in multiple settings. Successes reported both anecdotally and now in the literature in small series, in predominantly in liquid tumors, as you said, in acute lymphocytic leukemia, non-Hodgkin's lymphoma, and to some extent in chronic lymphocytic leukemia. Our first combination is with CD19, and obviously then it makes sense in terms of our own experience, in terms of the tumors we target in, having high expression of CD19.
CD19 is a molecule that's expressed on B cells.
So as I said, numerous second generation studies underway with lots of success. What has happened more so in adults than in the pediatric population to date, and I wonder if Bob or Jeff would comment on that, is more of a problem with cytokine release syndrome. We think from what we've seen that it seems to be tumor burden related, and if we can quantitate that appropriately up front and then do appropriate modifications to the regimen to manage that, that would be a potential way to avert that and manage it. The other way is to use anti-IL-6, tocilizumab, in terms of the antibody, and then use that appropriately as well in terms of the time-in, because again, there are concerns around what that may do to T-cell expansion. T-cell expansion itself may be a biomarker in some respects. If you can quantify that to the extent that what's the critical amount needed to achieve effect is another thing that we're really interested in. Both the biology of responders and non-responders is of enormous interest, and certainly once we get enough patients at a multi-site level, we'll be able to interrogate that further, because that is something we should have enormous interest in. The pediatric data just recently published in the New England Journal of Medicine had 27 of 30 patients respond, which is outstanding in pediatric ALL. And as I said, from a manufacturing point of view, there are enormous challenges, and it has been slow and careful going, but we have now enough to do both the clinical trial work and to support an initial indication. But as Bob and I were discussing last night, it really is the absolute form of personalized medicine, because you manufacture for each single patient one at a time. So I'll leave my comments there.
Let me just say a word about cytokine storm, because I don't think it's been defined yet. I think everybody knows, and not remembers, but knows about the 1918 flu pandemic, and which was particularly lethal for young, healthy people. And one of the reasons that was true was because that particular strain of influenza led to a massive activation of the immune system and an outpouring of inflammatory cytokines like IL-6 and TNF and IL-1. And that itself led to basically a collapse of the cardiovascular system and death. So when, in this instance, with checkpoint blockades or with CAR T cells, we are really activating the immune system to a great extent. And one of the dangers of that is inducing a cytokine storm, which is really what happened early on here and happened several years ago in a trial done in the UK, where one of these immune checkpoint activators was engaged and led to a couple of serious deaths with this trial. So let's define the CAR T-cell
I think we have
technology
a
little.
slide. Jeff has a.
Yeah, it'd be good to go back a little bit for folks.
So so if you can, it's the first time I'm seeing the slide, so let's work through it together. CAR is a chimeric antigen receptor. That's what it stands for. And what that means is this is a way of re-engineering T cells so that you don't rely on the inherent specificity of the T cell receptor. You just confer upon T cells the specificity that you want. And in this case, and in the case with the most experience, we use CD19, which is expressed by malignant B cells. And the way you target that is you use an antibody. So I know I said that everything, this is all about T cells, but actually the real trick, by the way, as an aside, that broke this field open was antibody technology. It was antibodies used to block checkpoint. It was antibodies used to decorate T cells. Antibodies are our friends. So you can isolate the part of the antibody that recognizes whatever target you want, CD19, and you confer upon that signaling modalities, signaling domains, T cell signaling domains, into one molecule. So the front part of the molecule is anti-CD19, and that's expressed on the surface. And the back end are T cell signaling modalities, and that's why it's a chimeric, right? It's two parts. And so it's recognizing tumors like an antibody would, but signaling the T cells like a T cell understands how to be recognized. Now, there was about 20 years of work that it took to figure out the best ways to do that, and there's still no consensus. But there is at least now a platform to build upon. And there were many choices. There were choices for what the target would be and how to construct that. There were choices for which signaling domains. And it was quite controversial. And actually, the one that went forward at Penn actually was a little bit surprised. Carl June used 41BB and not CD28, which was different. And those are then packaged. You then have to choose how you're going to insert that construct in a gene therapy way. So this is gene therapy. And there were many choices there as well. Many things have been tried. The leading technology that's been brought forward at Penn and now at Novartis is lentivirus. And there was any number of studies done that Carl June did in how to re-engineer T cells using lentivirus. The first experiments were in patients with HIV who are already infected with lentiviruses. So there was a great deal learned, years and years of learning, until those three things came together: CD19, 41BB with other signaling domains, and lentivirus. Those were put into autologous T cells, the patient's own T cells, and given back as a white blood cell transfusion, single dose. And what's different about this drug than chemotherapy and antibodies is it's synthetic and it's biologic. So normal considerations of dose and schedule and how to manage toxicities in relation to dose and schedule were suddenly completely different than we've ever known in oncology. So a very small dose given to a single patient, just 1 million cells, put that patient in complete remission. It was unexpected, and so toxicity is.
So
yeah.
I have to ask this question, and then I'm going to turn it over to the audience to seek questions from all of you. Some would argue that this is a boutique immunotherapeutic, that how many patients are we actually going to be able to take their own cells and expand them up and grow them, make them into chimeric antigen receptor T cells and infuse them in patients? Is this going to be accessible to individuals outside of developed countries, for example, or from patients who are living in rural areas. Is this realistic? Can we really gear up and do this? And I think, Steve, you have some thoughts about that.
Yeah. I'll try and address as best I can. We are committed at a global level, once we prove that it's safe and effective, to try and achieve as much as possible what you are questioning. I think there are parts of the process that Carl and Penn have developed that actually make it somewhat easier. So certainly in terms of the transportation that's needed for these cells, they don't need to be frozen, from my understanding, the way prior therapies needed to. And there may be easier ways to transport to facilities around the world that will have to be arranged geographically to make that doable, and then get back to patients. Why are we so focused also on the safety aspect is because we're going to have to develop sites that are able to do this in a safe manner, that are trained to do it, and then we'll be able to manage the toxicities. So currently, because of where we are, it's limited to places with bone marrow transplant expertise. And again, at a global level, you can argue where that can be done, including in continents like Africa, where there are, particularly in Southern Africa, places that can potentially do it. But it is something we at Novartis are absolutely committed to from an access point of view. And there are, one of the reasons we're so attracted to this particular therapy and technology that Penn had developed, there's some parts of the process that make it a lot easier to do than in times past in terms of transportation. I don't know if you wanted to comment on that.
I would say that it's a big concern. Is it a boutique therapy? And you have to be in New York or Philadelphia or someplace like that. It's absolutely the goal to make it available, to make it an FDA-approved drug that will be available. And if we just have to trust that if a drug works like this, we'll make it available. I think of my time as a fellow and learned from my mentors about how bone marrow transplantation was developed. And boy, it was boutique therapy when it started. It was in the hoods of people's labs long before regulatory agencies. And now it's an industry. And thousands and thousands of people have been cured with bone marrow transplantation. It's a cell-based therapy. So I think the analogy is quite real. It's 2014, so we're dealing with new types of toxicities. We're dealing with lentivirus and gene therapy. We can handle this. And it's not going to be 3 patients today, 20 patients tomorrow, and the world next spring. It'll be iterative. We have to learn the new rules of toxicity, dose, schedule, indication. But I think there's and the cytokine release syndrome, which has been understood because those patients have been so well studied, even at a molecular level, already with a therapy designed to treat that toxicity, speaks to the rapidity to which one can go from boutique to conventional therapy. And we have to remember, some of the therapies we had when we started, cisplatin was a pretty nasty drug when it first came out. And a lot of people still is. You can, the nightmares I can hear from some of the folks who educated me about giving cisplatin to the first patients were nightmares in terms of kidney toxicity. Now, it's outpatient drug used literally all over the world. So we can do it if the drug mandates that we learn how to do it.
Thank you. So let me turn it over to the audience for questions. Lots of more questions I want to ask, but I want to give everybody here a chance. Do we have questions? Is there? Yeah. Go ahead.
Hi, well, thank you all for a fascinating panel. So a question really for Michael and Steve around combinations. Jeff, in your second slide, you showed sort of the most promising survival curve with combinations, which was sort of tantalizing. But then later on in describing it, you accurately, I think, pointed out the complexity of it and how it could be different in different patients and at different time points even. So for Michael and Steve, sort of how are you starting to think about combinations and sort of which patient should maybe get which combinations? Obviously recognizing that's a long road, but how are you starting to go down that road and think about getting the right combinations to the right patients?
Well, it's a great question. I can tell you, as Laurie and others know who have worked with us, it's a question we ask ourselves every day. And it's through convergent evidence, none of which is definitive. So the approach that we take at BMS, and I think other companies take as well, is to look at preclinical data and the promise that of either additive or synergistic effects in animal models, knowing that that is neither necessary nor necessarily sufficient to predict the clinical experiment. However, it provides a little bit of direction so you can start to make some choices when you're combining at least IO agents. When it's then time to start try IO agents, we have developed through some of our folks in our early development space benchmark endpoints which we can hopefully rapidly make decisions on the basis of early clinical signals. Now that can be challenging because the field of immunotherapy has now rewritten the rules about what is the relevant endpoint to ultimately achieve survival. That said, a lot of ongoing work is being done to try and correlate early endpoints with longer term value, such as types of responses, depth of responses, with the potential for PFS or an OS. So we then do a clinical phase 1 experiment. We then do a lot of translational work from that experiment to try and inform in an iterative way whether in fact the preclinical data that led us to combine a LAG-3 with nivolumab, because we thought that T-cell exhaustion might be part of the reason why anti-PD-L1 does not work in some patients. That gave us the rationale for doing the experiment. The experiment is ongoing. We will combine the clinical results with the translational work and then make it will become iterative science, because there is no clear way of giving you a precise roadmap to combinations of IO agents. We have a philosophy about how to combine IO agents with non-IO agents, which is a Bristol-Myers Squibb approach, and I don't know what Steve wants to add, but it relates really to thinking about what patients in society wants. They want transformational therapies with higher quality of life. So we're looking to combine IO agents, and if you've ever seen a patient treated with immunotherapy, be it a CAR T-cell therapy or checkpoint blockade, and they would come into the room after they're completing their therapy, you would not know that they were necessarily receiving therapy, because their quality of life after the intense period of therapy tends to be quite good. So the approach is to look for, that we have, is to look for very high quality, innovative, latest generation targeted therapies to explore them with IO drugs.
Steve, do you have
Yeah,
anything?
quick additive comment. I think, I just want to credit the Food and Drug Administration for recognizing this area, in terms of developing draft guidance on how to develop 2 investigational agents, which I think has both enabled the field and allowed us to have, very fruitful discussions with them, and allowed actually fostered cross-company collaboration as well. So, that draft guidance is available, may remain draft for a long time, but it's very helpful in terms of how you can develop a path forward to get drugs registered. And then, the only other additive comment, we need Bob's mice to do all those experiments, but it's humbling, because once you go into humans, dose, schedule, and getting that right is sometimes time consuming. So the more we can do more preclinical iterative work in more relevant models, I think the more helpful the clinical scenario will be in terms of speed.
There doesn't seem to be a massive desire for questions, so you can ask more too.
Okay.
Sam Waxel. Jeff, you mentioned earlier, we've all sort of know the nuances that are important in T-cell recognition in tumors, and what happened treatment with immuno-oncology agents. And you get about 20 or 30% of the patients responding, and then the rest don't really respond. And those same patients get autoimmune sort of sequelae. Do you think that part of the reason that those patients respond are that they're the most foreign antigens vis-a-vis self-recognition, and the other ones are too much like self? And do you think that may be the future of cancer vaccines, trying to drive more foreignness, as it were?
So you're referring specifically to the checkpoint blockade stuff, not the CAR, because that's a whole different ballgame. But in the checkpoint blockade, again, I think that depending on the response or non-response, it could be 2 absolutely opposite reasons. It could be a lack of an antigen recognition, and so vaccines or some approach to driving, even radiation, things like that could be positive for some patients. But in many cases, the T cell had no problem recognizing an antigen. It saw it. It just got shut down because the environment that it saw that antigen in led to this allergy or exhaustion. So this is where the biomarkers are going to be key. Can we identify which patients have T cells? You know, there's work now coming out by groups like Mark Davis that suggests that you could really get a T cell almost against anything you'd want if you actually look carefully with markers like what they call tetramers to do it. So I think the first thing we've got to do is